Welcome to our Blueprint Neurotherapeutics Network for Biologics question and answer webinar for this afternoon. My name is Anne-Marie Broome, and I am the program director for BPN-Biologics, as well as other biologics programs within the Division of Translational Research at NINDS. So we welcome you for an informational webinar and look forward to your questions associated with this. So to start with, let's actually look at some of the parameters in webinar logistics. This webinar will be recorded so that you will be able to access it after the fact and be able to go back and review any information that you may have missed. The recording will be made publicly available from our website, and a PDF copy of the slides will be embedded with links and sent to all registered attendees of today's webinar. Please use the Q&A box to post all your questions, and we will answer them either live through the Q&A or after the presentation has been completed. Questions can be entered at any time and then will be read following the presentation for this. For project-specific questions, however, it would be best for you to contact us either by phone or email and send us a draft of your AIMS page so that we can set up a call to specifically answer those questions. So let's talk about BPN-Biologics and the framework of the program itself. And so BPN-Biologics actually is an expansion of the existing Blueprint Neurotherapeutics for small molecules, which has been in existence for a significant amount of time and has enjoyed much success. And the basis for this is really to combine the strengths of both NIH and industry expertise and aid in shepherding neuroscience drug discovery from PI-initiated projects. So we start with those NIH and investigator-initiated ideas and really count on the fact that we are dealing with NIH investigator is bringing the expertise associated with disease biology, as well as assays and model systems to validate and examine target engagement as well. NIH provides industry expertise in which we have consultants with extensive pharma experience across all research and development disciplines, and they are pre-established industry standard contract services available that have been vetted through NIH NINDS. The ultimate goal for the program is so that the PI will maintain their IP and develop it along the pipeline to a commercial exit strategy, as well as de-risk projects as they advance through the pipeline. And so looking at this, the ultimate goal and success of the program and projects is really to advance them to the clinic and hand them off to whichever of the strategies you plan to operate under. So this gives an example of what our program goals are more fleshed out. And so our program broadly accounts for different biologic modalities, disease indication, and entry points. And so to that aspect, we deal with large biomacromolecules, cell therapies, as well as gene therapies as a classic subset, as well as some other biologic modalities that are specified by IC. We provide non-dilutive grant funding and the necessary resources, which includes contracts, consultants, and expertise that are typically lacking in much of our research community, or may potentially be a hurdle to successful completion of some of these projects. Our real goal is to de-risk the potential therapeutics to the point that industry will invest in them, allowing potential new drugs to reach patients efficiently and streamline this process. So we want to identify the best ideas for translation through this funding opportunity and associated infrastructure, all the while preserving the PI's or / institution's intellectual property to facilitate licensing from start to finish. So our program is for both academics and small businesses. We have 2 separate PARs for each. A PAR 21-163 is for all applicants, both academics and small businesses, driven by PI. The PAR 21-233 is for SBIR eligible small businesses. And so one of the early decisions that you will have to make as a PI is to determine which of these FOAs you will actually apply to. Grant funding for the support of biotherapeutic discovery and development projects into the clinic is really the ultimate mission, and the opportunity to get access to NIH-funded consultants, contract research organizations, as well as expert NIH project managers and scientific officers for this. They will be reviewed using a multidisciplinary special emphasis panel review, which is designed to address the gambit of topics within your application and biologic. Current upcoming receipt dates are February the 9th, 2022. That's the next one. And then past that, August 9th, 2022 on a continuing basis for the next 3 years. Our BPN-Biologics program shares a trans NIH program with 9 participating institutes as shown here. And the reps are as shown and are your point of contact when you first think to examine or apply to our PARs. And I'm not going to go through all the particulars of this because this will be available to you, but as general information, it's very important that you as applicants talk to your IC reps from the previous slide to determine and make sure that your proposal is within the mission of the IC in which you are submitting your application. Oftentimes that involves discussions with NINDS personnel. So you will certainly be able to ask both us about BPN-Biologics infrastructure questions, as well as the IC to which you submit. Now, this is looking at how the BPN-Biologics Network operates. BPN-Biologics is unique across the fields in that it offers a customized combination of infrastructure, expertise, and funds. As a project PI, you will receive grant funding for biological activities, free access to our network of CROs, and a team of consultants and NIH staff who will assist in managing the project. For each project funded under this FOA, the NIH will assemble a customized lead development team, which we refer to as the LDT. The LDT will be co-chaired by the PI and a BPN-Biologics consultant, and will include members of your PI team, additional BPN-Biologics consultants with expertise in areas needed to accomplish your goals, and as well as NIH staff. The LDT's ultimate charge is to develop an overall strategy for the project. And this includes milestone proposals to help and studies to be conducted by BPN-Biologics contractors and coordinate the activities across different research sites. So ultimately, what ends up happening is you have a flexibility in the mix of contract access and grant support you require, from little, in which all the research is primarily done within your laboratory as a PI, or the other end of the spectrum in which you are requesting the majority of the work to be completed by NIH contracted resources. Overall, then, the PI's team, though, needs to retain the intellectual property within the PI's institution for this. So a word or two about inventorship, since we left off with that. So our intellectual property expectations are such that inventorship is determined and defined per US patent law. NIH Blueprint has no stake whatsoever in the IP, and therefore, prior to receiving a grant award, a PI's institution must have upfront IP licensing agreements in place with all potential investors, and they have to address who will hold the title to the IP on the new the biological matter use, as well as any royalty arrangements that are in agreement. These IP agreements should aim for unencumbered IP consistent with Blueprint program so that you can create a clear and licensable product for this. So you want to make sure that in your assembly and submission of IND applications that you are including support for this particular activity as part of your plan, right? So the NIH, as you have these particular meetings with the FDA undergoing this intellectual property evaluation, you want to make sure to include NIH staff and consultants on the LDT within those meetings so that everyone is on the same page. Now, looking at the program structure. Now, the basis here is for convention, we're going to be speaking in terms of the UG3, UH3. But if this is an SBIR application, then the UG3 phase is equivalent to the phase 1 of the U44 application. And the UH3 is equivalent to the phase 2 of the SBIR application. So the FOA is written broadly to account for these different modalities and disease indications, as I mentioned in a previous slide, but it also takes into account the fact that we have variable entry points. And projects can enter at either the discovery stage or the development stage, based on the modality and the level of associated risk. That results in different trajectories and different expectations on what's really needed to successfully move through the pipeline. So if you look at the discovery stage, it primarily encompasses activities associated with lead optimization, while the development stage is utilized to advance a development candidate through IND enabling toxicology studies in the phase 1 clinical trials. So what that means is that proposals need to be very streamlined to get through everything that must be done within the desired timeframe, because all projects should reach the clinical trial stage, regardless of the activities or the entry point, within a maximum of 5 years. So looking at the breakdown of what that actually means and how you break down these subsequent application guidelines. So for the UGUH3 is research cooperative agreement by mechanism. Its length is up to 5 years. The UG3 phase can be up to 2 years, with a minimum likely to be at least 6 months. The budget is not limited, but must reflect the actual needs of the proposed projects. So in essence, ask for what you need to accomplish the activities proposed within your application. The U44 phase 1, phase 2 aligns with our UG3, UH3, as I mentioned previously, where a phase 1 is up to 2 years and a phase 2 is up to 3 years. Now, there is a cap on budgets for the U44 mechanism, in which a phase 1 can receive up to $700,000 per year, where a phase 2 can receive up to $1.5 million per year, but must be justified as both reasonable and appropriate for the activities proposed. Now, this leads to the idea that even though there are 2 phases, the UG3, UH3, there will be go, no-go milestones established that must be met every year to transition to the next year of funding. So your LDT team will help to establish clear quantitative and definitive milestones. Having a good idea of these milestones up front while you're planning for your applications is a good idea, but not an absolute necessity for your application. Those will be established during the awarding period of a meritorious grant. Annual go, no-go points at the end of each year occur and are evaluated via an administrative and external consultant review. And so transition from a UG3 to a UH3, depending on which year you start, is tantamount to your success. And of course, there is an expectation that there is attrition of projects that go through and do not meet their milestones. So let's look at the discovery stage projects. So when looking at the entry criteria for a discovery stage project, you want to have identified one or more lead biologic agents that are sufficiently profiled so that the parameters still to be optimized can be quantitatively specified. The agent should show in vivo efficacy using clinically relevant outcome measures, for example, biochemical, anatomical, and/or functional outcome measurements whenever possible, and in vivo target engagement, right? So by example, that means measurement of target binding or proximal downstream effects at the clinically intended sites of action for this. So, and then from that, you want no obvious legal or IP constraints coming from that. The end goal of a discovery stage project is that you will identify a clinical candidate with the appropriate bioactivity, stability, manufacturability, and bioavailability by the intended route of administration that will naturally progress into the developmental stage, and then show that target engagement with a defined minimal and optimal dose by the intended route of administration and in vivo efficacy where applicable. Now, this is just a short list of some of the activities that are supported during the discovery phase. For instance, characterization, lead optimization, determination of the ROA, demonstration of adequate stage appropriate preliminary safety studies associated with this. So projects that meet this goal in discovery stage will enter the development in the UH3 phase, beginning preparatory activities. So you're ready then to enter into the development stage. Now, one detail here is that all applications proposing to enter the development stage will begin with a UG3 funded phase, even if that phase is not a discovery phase. And we refer to this as the preparatory development stage if you are not using a discovery start. Now, that UG3 funded phase can be up to 2 years in length at a maximum, and should be at least 6 months at a minimum in preparation. The entry criteria to come into the system for the development is a strong body of data linking the putative therapeutic target to the proposed disease indication, as well as a rationally laid out biological activity by an identified route of administration. And you want to make sure that you can demonstrate that the PI and the PI's institution are able to develop or commercialize the proposed biologic, because the end goal of the development stage is submission of the IND package and the initiation of the phase 1 clinical trial in healthy individuals, looking at safety and toxicity. So, for example, a list of the development stage activities are shown here, in which they can include development stage IND preparation activities such as non-GLP toxicology studies, formulation, scale-up manufacturing and stability, the repeat of an in vivo study in either the same or different animal model, and as well as it's encouraged that there be held a pre-IND meeting with the FDA if it has not already been conducted. The development activities further can include cGMP manufacturing for the phase 1 clinical testing, IND enabling safety and tox studies, preparation and filing of the IND documents, and the phase 1 clinical trial itself. When looking at the clinical trial then, because the ultimate goal is to get this into phase 1 clinical trial, is that you should examine the clinical trial design attributes. So for the clinical trial, you want to conduct either a single dose or SAD, single escalating dose, which may be a placebo-controlled or open label study, a MAD, a multiple ascending dose study, if your biologic has a short half-life. And the clinical trial outcomes may include safety, tolerability, PK/PD, target engagement, and target modulation endpoints. Now, BPN-Biologics contractors can conduct phase 1 clinical trials for investigational agents that may include large biomacromolecules in healthy volunteers or a demographic subset of healthy volunteers. The caveat here, however, is if only diseased patients are expected to be enrolled. The applicant's own clinical site must be proposed instead of BPN-Biologic contractors. So when planning your clinical trial component of your application, we strongly urge you to discuss your plans with us, NIH program staff, prior to submitting your application to determine whether a clinical trial is both feasible within the time frame and whether available BPN Biologics contract resources are needed. So, some general tips on preparing your application and planning for it. Number one, read the funding opportunity announcement very carefully, looking at details and instruction needed. Discuss your proposal with NIH BPN Biologics Program Director, myself, and or other NIH staff, as well as contacting and discussing it with your home institute's program director for disease interest. If you will be submitting an SBIR, then we will need to have you discuss with the SBIR program director for SBIR specific issues that must be addressed. When planning your application, put forth a solid scientific preliminary data dossier to support your proposal and address the rigor of that data in your research strategy section. Address any obvious criticisms of the biology, as you are the expert within that field. Clearly indicate what will be done as part of the grant and what is expected to be done of the BPN Biologics contractors. Spell it out very specifically. Within the FOA, it actually outlines a table in which you will identify the activities done within house of the PI or the BPN Biologics contractors. Make sure to include budget pages for all years of the grant. And I'll put a little asterisk here. A budget is not required for BPN Biologics contractors, only for work that will be conducted within your laboratory. So, make sure to also consider and address rigor. This is a very important portion of any application. When supplying preliminary and supporting data, make sure to explicitly discuss the quality of the data being presented in prior publications in a detailed manner. For instance, ask yourself these questions. Were these experiments done in a rigorous manner, utilizing randomization, blinding, inclusion, exclusion criteria, and the appropriate power analysis? That helps to get to the answer about how credible and strong your preliminary and supporting data is within the application. Within the approach, make sure to provide details for the controls being used for each type of experiment and appropriately highlight potential confounds like surgery exposure, genotyping, variability, human placebo effects, right? So the best offense, or wait a minute, I think I've got that backwards. The best defense is a good offense. So prepare wisely. Include details within the experimental design about the reduction of potential biases and include subsequent rigor experimentation, including the blinding, randomization, and inclusion-exclusion criteria that you use to evaluate your preliminary and supporting data. Describe the source of the data on which the sample size estimation is based and provide details about the analysis itself. So this is very important within the approach section of your application. So things to do in preparation. Number one, complete your required registrations at least 6 to 8 months in advance of any proposed receipt dates for BPN-Biologics FOAs. If you're applying to the UG3, UH3 FOA, and any proposed budget year exceeds $500,000 in direct costs, this excludes F&A costs, then you need permission to submit that application from your IC. These requests must be made 6 to 8 weeks prior to the submission date so that review and administrative approval can be made for an exception. During this time, it's also worthwhile to talk with your tech transfer and business development group at your institution and plan for funding patents and licensing activities, building in those timelines into your grant as well as budgets needed to accomplish those goals. Now, of course, because there are things to do, there are definitely things not to do. So do not plan for this award to be the sole funding for your laboratory. Milestone-driven programs notoriously can end abruptly and suffer from a high degree of attrition. Do not under-resource your budget to avoid any limits. Ask for what you need to accomplish the goals proposed within your application. Do not plan for BPN-Biologics contracts to be available for disease biology or one-off in vitro ADMET experiments. Do not code your application as a clinical trial if you plan to use the BPN-Biologics contractors. And if you do plan to run your own trial, it will be considered a delayed onset clinical trial for this. We have a variety of different informational sites that you can go to. This link actually directs you to the Blueprint Neurotherapeutics Network for Biologics, BPN. And on the side here, there are several child pages that link out to important information that can be utilized during preparation of your application. And I'll highlight one of these here, for instance, the BPN-Biologics frequently asked questions. Many questions can be addressed and answered early on by examining this FAQ child page within. Of course, for project-specific questions or follow-up to this webinar, you are more than welcome to contact myself at Anne-Marie Broome at nih.gov or our health program specialist, Elena Barnova, at nih.gov. Additional information can be found within the FOA itself, as well as our BPN-Biologics website for this. And so I'll actually stop here and look for questions from the attendees. So April actually said, what program supports the drug development before phase 1? So BPN-Biologics is a program that supports drug development before phase 1. If you are early in the discovery phase, then there are also programs such as Ignite, which are available. Do we have any other questions? We'll give it a couple minutes while people are typing. I know my typing skills are pretty slow. But I encourage anyone who would like further information to refer to our BPN-Biologics website, contact us either through email or by phone, and we will be happy to discuss. As I mentioned early on, also providing a draft of your specific aims can actually start a conversation with NIH staff and assistants. Let's see. Somebody indicated, can multiple staff from one company submit the SBIR at the same time? If the application is proposing activities, there can be no duplication of the application being submitted to SBIR, correct? So you can submit multiple applications, but the specific aims and goals must be clearly distinct and do not overlap for the application to move forward. Huang Kim, yes, you will receive the presentation slides PDF after this webinar is complete. All registrants will receive the slides. Okay, I'll give it one more minute for our typists. Can we choose the study section if more than one application is submitted? So that's a very good question, April. You do not get to choose the study section because the study section actually incorporates all of the applications into one special emphasis panel. The members of those panels are experts gauged towards that particular application. It really isn't a problem as long as the applications are unique and do not overlap if more than one application is submitted to a section. So we have seen this happen, but as you know, a mention here, be very careful that they are unique studies, unique disease subsets, and unique specific aims to address this. All right, Yuri Miller, can UG3, UH3 have a subaward with a small business founded by the PI? And other way around for U44, a sub to founders lab at the university. So, a UG3, UH3 can have a subaward to a small business founded by the PI. However, there has to be a clear delineation and justification of the activities to be supported by that subaward. So you would include that into your application. For the decision of whether you would submit to a UG3, UH3 mechanism or a U44 is one that should be considered prior to submitting an application, right? So the U44 has subsequent SBIR restrictions that must be obeyed, but yet the opportunities to be funded through the SBIR program may potentially benefit an application as a U44. Does that answer your question, Yuri? Thank you. All right. I'd like to thank everyone for attending our webinar on BPN-Biologics today. It was great having everyone here. By all means, feel free to follow up with us. We look forward to chatting with you soon. There will be another webinar series if you think of additional questions or points that will be coming in December. If you would like to pass on the information to your colleagues and associates. Thank you very much and have a good afternoon.